Analyses of the breast cancer microbiome. So kind of to just start off, the microbiome is really just the genetic material of all the microbes, such as bacteria, protozoa, fungi, and viruses that both live on and in the human body. And the microbiome is essential for human life, where the bacteria, for example, in the microbiome helps to digest food, regulate the immune system, and also produce vitamins. So moving to the introduction, breast cancer is known to be the second leading cause of cancer death in the United States, and it's known that breast cancers interact with local microbiota. Initially, there were earlier studies that looked at the human milk microbiota, and bacterial communities have been identified that are associated with the human milk microbiota, and later studies then looked at the microbiome of breast tissue, where these studies investigated the relationship between the memory microbiome and breast cancer. So now it's widely known that breast tissue is not sterile and human milk is not sterile. So my project is to do a meta-analysis of studies investigating breast cancer and the memory microbiome, where my studies of interest include a study by Urbaniak et al. In 2016, a Higgins study in 2016, a Schwann et al. Study in 2014, and a Chan et al. Study in 2016. And my project aims to address two questions. So first, are the results consistent with the original study? And two, do we learn something new from the reanalysis? So these are the results of the data 2 analysis of the data from the Urbaniak study, where these are the proportional abundance plots made through data 2 and phyloseq, and these are the phylum, family, and genus levels, where on the x-axis we're comparing breast tissue and healthy tissue, and then on the y-axis there's the abundance. So at the phylum level, the most abundant microbe is Proteobacteria, and at the family level it's Pseudomonadisiae, and at the genus level it's Pseudomonas. So these are the results that we found through the data 2 analysis, and these results are similar to the original study, but Pseudomonas is not shown. So these results are similar in that in the original study at the genus level, there was Acinetobacter, Anoxibacillus, Bacillus, Micrococcus, and Prevotella present, but Pseudomonas was not present at the genus level in the original study results. And these are the beta diversity and alpha diversity plots made through data 2. So in the beta diversity Ray NMDS plot, looking at the breast tumor and healthy samples, they do clearly diverge. So that shows that the samples are different and they don't mix together. And then the alpha diversity, the similar thing is shown as the breast tumor and healthy samples separate from each other. So on the left is the abundance plot from the data 2 analysis of the Urbaniak data using the Silva version 138 reference database. So on the x-axis, it is separated by each sample to compare with the graph from the original study, which looks at the breast tissue microbiota in 43 Canadian women identified by 16S RNA amplicon sequencing in the Urbaniak dataset. So at the genus level, in our abundance plot on the left, there is a clear abundance of Pseudomonas Prevotella 9 Micrococcus, as well as Bacillus and Anoxibacillus. And on the graph on the right, there is a clear abundance across the samples of Bacillus and Micrococcus, along with Prevotella in some of the samples and Staphylococcus. But there is no Pseudomonas present, so that is a very clear, significant difference between our results and the results that they found. So for the Schwann et al. Study, these are the results from the data to analysis of the Schwann data, where these are the proportional abundance plots at the phylum, family, and genus levels. And at the phylum level, these results are very similar to the original study, as both studies have Actinobacteroida, Bacteroidota, Firmicutes, Proteobacteria, and Microbiota. And they are similar at the genus level too, as both have Methylobacterium and Sphingobium present. However, Escherichia shigella is not present in the original study's findings. So my next steps are to complete the phylogenetic and principal coordinate analyses for the different data sets, and also to conduct a meta-analysis by combining the results and output of all the studies. Additionally, I also want to run statistical analysis on the combined studies, such as multivariate analysis, and also random forest application. Thank you for watching my presentation.